two objectives here. The first is to understand the benefits and limitations of a few different bioinformatics databases. And the second is learning where and how to best access the data stored in these databases. The three main ones we're going to focus on are GTEx, GEO, and TCGA. So let's get into it. The first is GTEx. This stands for the Genotype Tissue Expression. You can get to the database through this website, the GTEx portal. So this database is a comprehensive study of tissue-specific gene expression and regulation. This is looking at non-disease tissues. So these are control individuals. And there's 50 different tissues covering 1,000 different individuals. And a lot of the time, it's the same individual, I think, contributing different tissues. In this database, they have whole genome sequencing, whole exome, and RNA-seq. The bulk of it will be RNA-seq. But it's open access data to gene expression, QTLs. So this is correlating variants from the whole genome, whole exome sequencing with expression and histology images. So this is sort of a breakdown of how they collected this data. They had many different donors. They took samples from many different tissues, some from the same donors, performed sequencing from all of the different tissues, and then did expression correlation with SNPs variants to see whether having one allele or another leads to a different expression. They store all of these samples in a biobank. If you have a research question that would be answered by actually getting the tissue sample, I believe you could go through some hoops and actually get some of that tissue. And then they store it in a database, which you can access. This is showing probably mostly all of the tissues and how many samples were collected for each tissue type. Some tissues are better represented than others. And I believe the color versus grayscale indicates how many of this tissue has DNA eQTL information, and then the gray samples would only have expression. This lists out the types of data that they've been collecting over the years. It just started with expression, and then they expanded to QTL analysis, and now they're even doing single cell and some ChIP-seq methylation data, it looks like. But the focus and the main source of information thus far has been on expression data. So that's going to be the richest amount of data that they're going to have available. On the website, you can do a multi-gene query to kind of get a snapshot of how the expression of a few genes of interest may look across all of the tissues and get an understanding of how tissues cluster based on these genetic profiles. You can see on this example query, a lot of our brain samples cluster together. And then there's other clusters of tissues which are less obvious, but this is an interesting way of getting a snapshot view of a few genes of interest across all of the tissues. This is another view. If you have one gene that you're really wanting to look at, you can get the bar plot, violin plot distribution of this one gene for all of the tissues. So you can get an idea of, for P53, how is the expression look like in each one of these tissues? Is it going to be easy to capture the expression of this gene in the tissue that you're interested in? Is it going to be more difficult? These are questions you can answer looking at this type of data. So why would you want to use data from GTEx? If you want to understand expression of a gene or a small number of genes in normal tissue, if you want to understand how this expression changes from tissue to tissue, do you want to make a justification for wanting to study your gene in a very specific tissue of interest compared to taking a blood sample and looking at the expression in that tissue? If you're going to compare a biologically relevant tissue to something easier to capture, like blood or some sort of other specimen, you may want to use GTEx. It's difficult, but you may be able to use GTEx to provide a set of normals for your comparisons to a disease of interest. I know there are groups out there who have developed ways to use GTEx as a control for the cancer data sets that we're going to talk about later. And I've listed some of those publications down below. The main issue is they're different data sets and they're each going to have their own biases and batch effects. And one platform is different from another platform. And it's really hard. And then the last thing is GTEx is unique because it has the availability of eQTLs, the expression QTLs relating variant SNP changes to expression. So we can make some sort of assumptions about regulation, how different genotypes can be related to expression. And you can look at that across tissues and across individuals. It's a very rich database of information. The second database that I'm going to present today is the Gene Expression Omnibus, also called GEO. So this is a public genomics data repository of all types of array and sequencing data. Usually, your first experience with GEO will be that you've done an RNA-seq experiment and you want to publish it, but then the journal says you have to provide the raw data in a public manner, and they suggest to use GEO. So you package up your raw expression data and some information about your samples. You can upload it to this website, and it will be available for anyone who wants to use it. It's arranged in an organization of platforms, so the sequencing machine, into samples and into series of analyses. And then through this, you can do quick visualizations, look at expression trends and changes. So GEO has a really nice setup for filtering. You can search by a number of criteria, including keywords, limiting it to certain species, study types, if you want it to be limited to only NGS, next generation sequencing, or if you want to limit it to microarrays, you can do that. You can subset it based on sample characteristics such as age or samples with some certain disease. You can look based on sample size if you want to find studies with a lot of samples, or even search for a study done by a certain author. You can filter based on all of those things. Here I've limited the study type to expression profiling by high throughput sequencing. Just for an example, I'm showing we're limiting to smoke wildcard. So this will capture any sort of smoking or smoker. So we're looking for something related to smoking. Also, keyword diet, we're limiting to mammals that are not human. So this will be mouse, rat, any sort of species that can be studied with diet and smoking. So I got one example. This is the information about this analysis. It's a specific series, and this series has a number of samples. And we know this was the platform that it was sequenced on. It's a mouse data set, and there were 12 samples. Some of them have different types of diet. And if we click on one of these samples, we see the listing for that particular sample. We get an idea of how the mice were treated, how the RNA was taken from the mice, how it was sequenced, and then some information about how the sequencing was analyzed. It was mapped with STAR with this particular configuration. They used feature counts to generate the counts with these options. They filtered the normalized expression, and they mapped to the MM10 mouse genome build. So this is important information if you want to use this data in any kind of analysis. This particular data set gives us raw counts. Normally, Geo asks you to upload the normalized expression. So if you want to do true differential expression, you need the raw counts, which it makes this listing really nice. But if they only provide normalized expression, you'll need to download the raw sequencing FASTQ files so you can align on your own and generate raw counts, which you will need to do a differential expression analysis. Now, to get that FASTQ, there's a way to download it directly from the website. If you see right here in the information down below the samples and the supplementary file, there's this SRA run selector. You click on that, it will bring you to this page with a listing of all of the samples. You can click on one of the samples, it will take you to this page, which is a sequence read archive listing for that sample. You click on data access, take you to this page, which will have a link for that particular FASTQ file for this sample from this project. It's a process to click through each one of these. So if the study has a lot of samples, I don't suggest using this click through download from the website method. An easier, but possibly harder to run method would be to use OSC, the Ohio Supercomputer. They have stored as a module the SRA toolkit, which you can, this is a screenshot from the OSC website about the SRA toolkit. You load the module, you run prefetch on, I assume this is the study ID, and it will get a listing of all of the samples in this study. And then you can run as a job a script which will download all of your FASTQ files. It's the FASTQ dump command, which will download all of your FASTQ files for you. They warn you in this screenshot that you may have errors, and just talk to OSC and they should help you troubleshoot to get this to work if you want to go this way to download some data to use for your study or your analysis. Built into the Geo website is an application called Geo to R. It's a built-in R application. It's an interactive tool that allows you to compare simple groups of samples. Now, the groups have to be defined in the experimental conditions and the listings of Geo. Sometimes there will be experimental conditions that are not listed as specifically as characteristics, like all of these. You could use these through the R built-in analysis package. They have to be in the conditions list, and it will allow you to export a table of differentially expressed genes and do some visualizations based on that differential expression analysis. So this is for a single gene. It's showing the expression for one group of samples compared to another group of samples. This is showing, the next plot is the volcano plot of all of the genes in this comparison. There's a group of them in red, which are upregulated based on fold change, and a group of them in blue who are downregulated based on fold change. And all of these meet an adjusted p-value cutoff. There's a Venn diagram showing 3 different comparisons of groups in this data set and how the genes picked out with a certain significance cutoff overlap between these 3 different comparisons. And then finally, this is a UMAP plot showing the relationships between samples based on their gene expression profiles. It's a lot like a PCA plot. But all of these analyses are built into the GO2R application. So if you have a simple query, such as comparing 2 different experimental conditions, you could do that directly within the GEO website. So when would it be a good idea to use the gene expression omnibus? If you want to find studies that are related to your own study, which may support or not support your own findings, this would be a good way to go. If you have a simple comparison to make, you can use the built-in GO2R group comparisons to check a gene out. Or if you want to download expression counts or raw FASTQs to make your own differential expression analysis, you could do all of these things through GEO. And also, if you have your own data set and you want to make it publicly available, you can do that through GEO. Okay, so the last data set database we're going to look at today is TCGA. This is the Cancer Genome Atlas. They've characterized over 20,000 cancer types, most of them containing matched normal samples, with 20,000 cancer samples with cancer and normals spanning 33 cancer types, not 20,000. Okay, so 33 different cancer types, each with a very large population of people with this type of cancer, and typically containing a matched normal sample as a control. It has a very rich phenotype information associated with each one of these cancer types. So there's a lot of information about cancer stage and what types of treatments they've had, and many things that you can use in your differential expression analysis. They say they've generated over 2.5 petabytes of genomic, epigenomic, transcriptomic, and proteomic data. So there are a lot of different data sources built into this database. There are different versions of the analysis on TCGA. I'll start at the bottom here, the legacy TCGA data. This was the initial data that was collected and published by the TCGA Research Network, and it was the first analysis through. Later, they added more samples, they updated their analysis programs, and published the Genomics Data Commons version of TCGA. And this is the reanalysis from the latest publication. Then there are 2 other types of analysis: the TCGA data in the UCSC RNA-seq recompute compendium TCGA. That is a mouthful. It's been co-analyzed with GTEx data. This is what I was talking about before. Some people have used GTEx as their pool of normals. And so this research compendium has developed a specific method of reanalyzing all of the TCGA and GTEx data together to kind of correct for batch effects and use GTEx as a normal pool of samples to compare tumor versus normal. And then finally, these have not been in order at all, but finally there's the Pan Cancer Atlas. They say it's the most comprehensive cross-cancer analysis to date, but it combines all of the cancers into one single analysis. So cancer to cancer differences can be assessed. And they've just sort of streamlined it, made it all uniform. And so that's going to be the best, most represented analysis of the data. Then there are different levels of analysis. So what you're going to see in most of the public databases will be level 3 data. This is going to be gene expression, counts, mutations, CNV calls, aggregated, and directly usable. If you want to get raw data or something in between, you have to request an account through ERA, Commons, and dbGaP, and go through a process to get access to those more raw levels of access. And there's a process to do it, but then you'd have to download big files and reanalyze it all on your own. For most applications, the level 3 data will be sufficient. So that's what we're going to talk about today. The final thing to discuss is how to access the TCGA data, because there are several different portals. They all access the same thing basically, but there are different ways of getting at it. So one of the ways is through one of the R packages. One of them here I've listed TCGA biolinks, and it's through Bioconductor. You can install the library, follow the vignettes, but they have a setup way of defining which TCGA database you want to pull from, which cancer you want to study, what type of data you want to download. And it downloads them all as R objects, which makes it easy if you're going to do all of your analysis in R to just download the data and analyze it directly. But it's a little hard to modify for your own use. So I don't use it, but I've put together guides on how to use it, but they have a really nice website for it. Another possibly more user-friendly option is CBioPortal. And the benefit to this access point is they have TCGA, they have the different analysis versions. So Firehouse Legacy was that initial analysis. This Nature 2012, I would assume, is the GDC. Then they also have the pan-cancer analysis. And they have slightly different sample sizes and slightly different phenotype information. And then the other thing is there are a lot of non-TCGA data sets here. So this is colorectal cancer. We have these 3 TCGAs, but then we have data sets that are from other groups. And here's one that even has over 1,000 samples, which that may be of interest if you're studying colorectal cancer. So, but we're talking about TCGA here. We click on one of these and we go to the listings for this is the pan-cancer analysis for colorectal cancer. There's going to be a GUI interface to look at different genes, look at the survival in this data set. There's a lot you can click and choose. If you want to limit it to only colon or only rectal, you can do that. You can look at different profiles. You can look at copy numbers or expression. There's a lot to sort through here. But if you want to download everything, all of the clinical, genomic, mutation, gene expression, CNV, you can copy the link from right here or just download it directly. I like to copy the link and use wget to download into OSC because I can genzip it, untar it there, and see all of the text files, all of the data, all in one place. So that's how I suggest getting that data if you're wanting all of the different types of data for one cancer type. I think my favorite way to access the TCGA data is through the UCSC Xena portal. Here's the website for it. They have a really nice option for visualization. You can choose the TCGA study you want to use. You can pick out a couple of different genes to look at the expression or the mutation or the CNV. You can select some phenotypes, and then you can do visualizations. Like if you want to look at the expression of P53 broken down by cancer stage, you could do that just really easily and get an idea of, is there a relationship between this gene expression and severity of cancer? Or is there a relationship between this gene and tumor versus normal? You can do all of that in this simple little visualization application in the Xena portal. So this is an example. I think it was one of Xena's examples, but this is the TCGA target combined with GTEx. So these are all of the samples. Each block, it says, is about 1,000 samples. This is telling us half of them are GTEx samples and half of them are TCGA samples. It's showing us the gene expression, the transcript expression of 3 different transcripts of this FOXM1 gene. And then this is the expression of a few other genes from one analysis of this TCGA combined with GTEx. So you can see there's a definite trend in this gene comparing GTEx to TCGA. So TCGA samples have a much higher expression of this gene compared to GTEx. And comparing these 3 different transcripts, you can see that it's mainly being driven by this 3rd transcript right here, which has the strongest difference between the 2 groups. Again, it's really high in our cancer samples and really low in our normal samples, where the difference is, it's kind of trending that way, but it's not as strong for these other 2 transcripts. It's this one transcript just driving what we're seeing with the whole gene expression right here. So that's what we can see in this simple visualization viewer with Xena. Then if you're done just exploring a few different genes, a few different phenotypes, you can download all of the genes and all of the phenotypes. If we go to data sets, this is listing all of the data sets in the Xena browser. It's listed in this website. So you can see, this isn't all of them, but this is all of the legacy TCGAs, and then all of the reanalyzed GDC TCGAs. And then these targets are the TCGA combined with GTEx possibly. And then here's the GDC pan-cancer data set right here. That is the combination of all of the TCGA cancers analyzed in a consistent manner. So I've clicked on one. This is the acute myeloid leukemia. Its code is LAML, and it's telling us that there are 15 different data sets of different data sources. So there's copy number, DNA methylation, gene expression, here's mutations down here. The phenotype is broken down into all of the phenotypes, and then the survival data. They usually just give us overall survival. There's CBioPortal. They have a couple of different survival events, more than OS. So you usually have PFS and RFS sometimes. But let's focus on gene expression. And you see there's 3 different listings here. The HT-seq is the program they used to get the gene expression. So at the base level, they have HT-seq counts. So this will be the raw counts. We get straight counting reads on a gene. And then they have 2 different normalizations of the counts. FPKM, fragments per kilobase per million reads, and then the FPKM, which is upper quartile normalized. And it's this FPKM UQ that you'll see most often in those Xena visualization plots that they do. But for our use, if we're doing differential expression with DE-seq or EdgeR, we're going to want to download the raw counts, because those differential expression programs, they make assumptions based on having raw counts. So that's definitely what we want to download if we're doing differential expression. And then I suggest downloading phenotype data and survival data too, if you're intending to do any sort of survival analysis. So if we click on the HTC counts for this cancer type, it's telling us there are 151 samples. And it's telling us that the unit of the counts is in log 2 count plus 1, which makes it a little difficult to analyze counts. So when we actually do analysis, we have to transform the log 2 counts back into counts. So to do that, we take this value, take 2 to this value, and then subtract 1, because we have to get rid of this 1 that's been added in. And then we round it to 0, because we want a raw count that is just a count. It can't have any decimal points at the end of it. So you'll see that in the hands-on activity that we do later, that I'm converting these log 2 counts into just raw counts. But this is the download page for the gene expression raw counts. You can download it directly onto your computer, or you can copy this link and download it into OSC or any sort of Linux computing environment you have. Also download the probe map, which will allow you to convert your ensemble IDs here into actual gene symbols that will be useful for anyone looking at the gene. But then there's just an idea of what the sample names look like and what the gene names look like down here. So looking at these sample names, we have to talk about that a little bit. So each TCGA sample is a combination of barcodes. So the first 3 parts of the barcode make up the sample ID. So sometimes you'll just see that sample ID as the barcode for the patient. Then sometimes you'll see more parts of the barcode if it's relevant. The main part you have to be careful with is this sample part. The 01 tells us that this barcode belongs to a primary tumor. Different codes will be defined by different definition types. So 01 will be primary tumor, 11 will be normal sample, and then anything in between, I think 6 is recurrence, and here, 2 is recurrence. I think 6 is metastasis, but they mean different things. And then the letter after this code tells us which vial the sample came from. So some subjects may have multiple samples that were pulled from them. And I typically take the vial A as being the best sample, but that might not always be true. But there's always more vial A's. Everyone should have a vial A, but some samples will have a B or a C. But in the hands-on, I filter for primary tumors, normal samples with a vial A. So when do we want to use TCGA? The biggest time is if you want to study cancer. And it's very particular types of cancer. So if you have a question, about a particular type of cancer and you want to test out your hypothesis or confirm findings that you've already seen in your study in cancer, then you can use TCGA to do that testing. If you want to download all of the different data sets, I would use cBiolinks to get the CNV mutation expression phenotype data all in one pull. If you want to download specific types of data, like if you're only interested in expression, then you can download that and the phenotype data pretty easily with Xena. And then you can use Xena visualization to just hand check a small number of genes for group differences for survival and get immediate plots to kind of see whether your idea makes sense. So that was all we have to go over today about bioinformatics databases. The next class session, we'll go through an example that I've downloaded from TCGA where we're going to do differential expression on primary tumors versus normals and see what we can get out of it. So if you have any questions, this is my email, and have a great day.